In the United States, dams have been a ubiquitous feature on rivers for the past 200 years. Numbering in the hundreds of thousands, these dams are used for flood control, hydropower, and many other purposes. Recently, however, advocates for dam removal have been successful in their efforts to free rivers from dams of all sizes. One such dam removal occurred in December 2014 on the Fall River in Gill, Massachusetts, a tributary that enters the Connecticut River just downstream of the Turner's Falls Dam. There, the Massachusetts Division of Ecological Restoration, the U.S. Fish and Wildlife Service, and the organization American Rivers joined forces to remove the International Paper Company Dam No. 2. This dam measured 30 feet long and 8 feet high and was built in the late 1800s, and it was the only dam on the Fall River. Removing it opened about 40 miles of habitat to fish looking to swim upstream to spawn. Hi, I'm Spencer Weinstein, and I work with Andrew Whiteley and Allison Roy studying fisheries conservation genetics at the University of Massachusetts Amherst. Dam removal results in a clear visual change to an environment. Rivers that were turned into reservoirs are once again allowed to flow freely. Stream channels that were narrowed or drained are restored as natural flows resume. However, changes to fish assemblages and biotic communities in these rivers are not as visible. To determine the effects of dam removal on fisheries, genetics is a crucial tool. My thesis examines, through use of genetics, the amount to which dams are a barrier to movement. Prior to the 2014 dam removal, a team of scientists went to the Fall River and collected caudal fin clips from Slimy Sculpin, the most abundant species, from 350-meter reaches above the dam and 450-meter reaches below the dam. Last month, I, along with my advisors and others, returned to the site of the dam to collect post-dam removal samples. Every sculpin was identified with a species code, measured, and assigned a unique ID, and a caudal fin clip was taken. In total, nearly 400 unique samples were collected. I have extracted DNA from these samples, and specific regions of repetitive DNA, known as microsatellites, will be amplified using PCR. From there, genetic information will be gathered, microsatellite loci scored, and the results analyzed. The analysis that I'm using, called sib-split, uses young-of-year sculpin identified through length frequency histograms to identify movement through the dam both pre- and post-removal. Through genotyping, individual sculpin are assigned to full sibling families. The presence of full siblings on either side of the dam indicates movement. By using data from before and after the dam removal, I can determine to what extent the dam was a barrier to movement. Humans depend on fisheries for recreational angling and in some communities to make a living. For many indigenous groups, a productive river is a mark of cultural significance. Dams have long been viewed as symbols of industrial power, but the times are changing. With local activism, rivers are once again being restored to their natural states. Through it all, it is important to understand the effects that these dams and their removal have on the ecosystem and on fish communities.